Thank you and welcome everyone to this week's Rough Cut seminar. So the weekly seminar hosted by the Montana Institute on Ecosystems, highlighting research on ecosystems and the environment from different disciplines. So I encourage you to go on to our website and look at the entire schedule of seminars we have for the semester. The other announcement I'll give before we get started with this week's seminar is this Friday is the first Friday of the month, so we're having a happy hour over at Bridger Brewing from 5 to 7. So please come on over and network informally over a, I guess we don't say, we're up right. All right, today we're very happy to have Seth Walk come and give this week's Rough Cut. He's an assistant professor in the Department of Microbiology and Immunology here at Montana State. He's been here since 2012. He has his PhD. He has a dual PhD in zoology and in ecology, evolutionary biology, and behavior from Michigan State. More impressive to me is that he has his bachelor's and master's from Penn State, where he was the drum major of the Blue Band. And if you don't know, that is a very big deal. So he's done postdocs at University of Massachusetts and University of Michigan. His lab here has, he's working with PhD students and undergraduates. He has projects funded by NIH and the Bill and Melinda Gates Foundation. And he's been doing great things here. He has almost 40 journal publications already. We're very happy he's here at Montana State. And we're very happy that he's here today to talk with us.
Great. Thank you so much. Thanks everybody for coming. Looking through the schedule of talks, today's talk might be a little bit different. I don't normally study ecosystem scale processes, but hopefully I can relate it back to some environmental issues that are kind of near and dear to everybody. So if you have to leave early or you want the punchline first, I think this is it. We'll get into a little bit of the background on E. coli and Escherichia in general, but these bacteria are often known as being colonizers or inhabitants intestinal tract. We can come back to this at the end and maybe have some more discussion about it. So just the outline for today, we'll talk a little bit about Escherichia, the ecology of these bacteria, their natural history. I'll give a little bit of background on population genetics, not a whole lot, but just enough to maybe set the scene on how we, or how our lab in particular has approached E. coli in the environment, and particularly with one concept called the clone concept. This sort of underlies or gives us a theoretical base for looking at different lineages of E. coli. my doctoral mentor's lab at Michigan State when I was and some of that work that continues here on campus. And then hopefully at the end, I'll give you a couple of punchlines about why this is at all important for mankind. Okay, so Theodor Escherich is the German pediatrician who Escherichia, the genus, is named after. About 1885, Theodor was looking at neonates in the hospital, getting stool samples from these young children and using Christian Graham's stain, which was en vogue at the time, to differentiate different microorganisms in basically the first stool samples of life, often called the meconium. So the main technique of the day, still used today, is microscopy. So Graham-stained slides, looking under the microscope. There's some very beautiful pictures in the notebook of Dr. Escherich at this time. And then he isolated these organisms on solid media and sent them to other investigators, other microbiologists in the field. And in time, one of these organisms that he discovered was named after him, Escherichia coli. So this is an early picture. This was about 1900, where this picture was rendered. This was by an artist at the time, just showing that the E. coli there on the right-hand side really went around the microbiology field. It became very famous very quickly. And so we've been studying E. coli for over 100 years, about 120 some years now, but we still understand very little about its ecology, how it makes a living, what essential nutrients are available, which ones, which processes or functions they might serve in different habitats. And so this paper came out in 1983. It's cited by many because it came up with a sort of understanding of E. coli ecology or framework. And this first statement is just to point out that E. coli is perhaps, maybe not perhaps, maybe it is the most studied organism on our planet. And so many different fields of science use E. coli and study E. coli. And so it's fragmented, our understanding about the ecology of this organism. And so concerted effort needs to be done to sort of bring these fields together. But Savago at the time defined a primary habitat and a secondary habitat. The primary habitat was the gut of warm-blooded animals. And the secondary habitat, this is sort of a direct quote, water, sediment, and soil. But if you read this paper, it's really anything other than the warm-blooded animal gut. So it's really, it's really kind of nebulous about what these secondary habitats are. But in general, E. coli, after coming out of the gut, has a negative growth rate. In some of the half-lives of water in sediment and in soil are listed there, about a day to 5 days. And these numbers have held up pretty well with rigorous testing. Transmission of E. coli, how we understand this or how this organism gets around, comes mainly from pathogens. So E. coli, as well, about 99.9% of E. coli are harmless to us, but that little percent that does cause disease causes a lot of disease and is very well known, primarily because of the symptoms. So traveler's diarrhea or ETEC, enteropathogenic or EPEC, they cause a lot of infantile diarrhea and death. So these are large sources of morbidity and mortality in developing countries. Then there's Shigella and enterovasive E. coli, enterohemorrhagic E. coli or bloody diarrhea. that'll catch your eye if you get that. So these diseases are common in the media. They're common to most mankind around the world. And so we understand E. coli in the context of these bad bugs. And primarily, we think of E. coli being transmitted through the fecal-oral route. This is the actual quote from CDC, through the fecal-oral route, primarily via contaminated food or water. As an epidemiologist, that's a little disconcerting because fecal-oral versus foodborne versus waterborne are very different routes of transmission. But the only fecal-oral route bacterium up here is probably the Shigella or enteroinvasive E. coli that are known to cause large outbreaks in like daycare settings and person-to-person contact. Other pathogens like enterohemorrhagic E. coli have foodborne vectors like undercooked hamburgers and raw spinach and leafy vegetables. Okay, so the routes of transmission then are defined primarily by pathogens. And we really don't understand how non-pathogenic E. coli get around. Okay, so E. coli has been used for many, many years as a fecal indicator, well over 100 years. And so the rationale for this is that they primarily live in the gut of humans and warm-blooded animals that defecate. And they have this negative growth rate in the outside environment. And some of them are pathogenic. So if you see some, that's an indication that there's a possibility of pathogens being present. And they're also easy to culture and enumerate. A little bit of history on this. Around the time Escherichia was defining these under his microscope, there were microbiologists in London looking at microbes in water. After E. coli became in vogue, sanitarians began to enumerate them from water. And in 1907 or so, it was suggested that E. coli, because it's such a commensal organism, is more prominent in fecal versus water sources compared to other coliforms. Okay, so the word coliform actually came from Escherichia's original description of the organism. So very early on, we began to associate E. coli with this fecal indicator application. So since then, there's been other named species of Escherichia. Coli was actually formally named after Escherichia in 1919. You can see these other species names. There's been some revision of this taxonomy based on molecular techniques and phylogenetic tools. A-decarboxylate, Lade, and Bladier have been renamed into other genera. Hermione. Knew I was going to do that. Hermione and Vulneris, not related to Harry Potter, are still related or nominally in the genus. So they remain Escherichia, but molecular evidence from housekeeping genes suggests that these actually are outgroups from E. coli, other Escherichia, and Salmonella. So this phylogeny does not support their current taxonomic position, nor does 16S sequencing. These are full-length sequences from the Ribosomal Database Project, and you can see that both of these fall out from the Escherichia cluster. So these are probably not Escherichia, so there's something else that haven't been named yet. So basically we're left with three species, coli, Fergusonii, and Albertii. Now Fergusonii and Albertii were both discovered because they were pathogens. Fergusonii causes some infections in the hospital, and Albertii was found to cause diarrhea in young children in, I believe, in India. So we start with the pathogenic identification, and then we move on to maybe some other members that are closely related. So population genetic tools are very important then to look at and to characterize E. coli because it gives us a better idea how they're related and how they might get around, some of the habitats that they circulate in. These are early studies by a man named Robert Philander. I think they span from like 1964 to like 73, I think, 1973. Anyway, at the time, Solander was using enzyme electrophoresis to understand population genetic diversity of a number of macroorganisms. So anything from birds to elephant seals. And these tools really help to understand the evolutionary ecology of these organisms. Just as a brief outline of what these techniques look like, this is a potato starch gel. So if you took potatoes and mashed them up really well and made a gel out of them, cooked the starch down and poured it and made a gel, you could slide in little wicks of paper that held lysate, either from plasma from macroorganisms. You can do this with plants, insects. But the idea is that these enzymes in this gel, once electrophoresis is applied, these proteins will migrate based on charge. And so you can separate allelic variants very precisely in this non-denaturing gel. Okay? So unlike diploid organisms like we are, that might have different alleles at the same locus, haploids have only one band here. So every A, B, and C is a different enzyme, and their migration pattern corresponds to the different alleles. Okay, so you can get a multi-locus genotype just simply by characterizing the migration patterns of the different housekeeping enzymes. Everybody there with me? Okay, so this is just it's a very nice way of looking at population genetic diversity. So Roger Milkman came out in Science in 1973 with the first population genetic study of bacteria, which was with E. coli. And he looked at 5 of these enzymes in about 800 strains. And he was interested in whether E. coli populations were structured based on recombination, so like higher organisms, or whether they were structured by some other mechanism like hypermutation. Something like that. So what he found was that there was the level of polymorphisms or changes that you would expect was less than the observed level of polymorphism was less than what you would expect given the population size of E. coli and the diversity in those individual alleles. So basically all this did was set the scene and say we can do population genetics on bacteria, and E. coli, Milkman concluded, was very highly recombinogenic. And the reason he suggested that is because when he looked in a single individual, or E. coli from a single individual, he didn't find a lot of variability. And so the explanation was there's a lot of recombination that was purging variation from the population. Okay? So that was his take in '73. Solander's lab then switched from macroorganisms to microorganisms about this time. And they reevaluated this, this conclusion by Milkman. Instead of looking at 5 enzymes, they looked at 20, and they looked at only 109 isolates, but some of these isolates were the same ones that Milkman had already published on. And Solander reasoned that if alleles were distributed at random in populations, then the likelihood of observing a genotype, which is a multi-locus pattern, was astronomically small. And you can calculate this just by looking at allele frequencies. So the most common allele frequency at each of the 20 loci, that's the expectation in the population. And out of these 109 isolates, they only saw 98 distinct genotypes. So what did that say? Well, they saw some genotypes more than once, and the likelihood of that event was very, very low if alleles were just out there at random. Okay, so there's linkage and there's clonal lineages that are persistent. Saw 98 distinct genotypes. So what did that say? Well, they saw some genotypes more than once, and the likelihood of that event was very, very low if alleles were just out there at random. Okay, so there's linkage and there's clonal lineages that are persistent. And the take-home here is that these unique phenotypes or genotypes that they saw more than once, one of those was in the lab rat strain K12 that was isolated in California about 50 years before this study, and that matched a genotype from an E. coli from an infant. So not only are these clonal lineages, but these clones appear to be long-lasting in the environment or in nature. Okay, so this really helped define E. coli populations as these clonal lineages, where recombination is rare. It happens, but the rate is about the same rate as mutation. But because of the isolation of these lineages, it's thought that they evolve changes rather slowly. Okay, so this is called the clone concept. And what it does for us is it allows us to go out and look at where these clones are and how they make a living in that environment. Okay, so one of the interesting things is within a host, if you go in and look at the E. coli there and you genotype them, you find resident strains and transient strains. Okay, so transient strains are shown in this table, resident and transient, but these are different dates. So each column here is a different date where a stool sample is taken from this one individual. Each row corresponds to a genotype, okay? So you don't really need to see the numbers, but the numbers are the occurrence or the frequency of those genotypes. But suffice it to say that some genotypes were seen once or twice and then gone, never seen again, where other genotypes, highlighted here in this box, were seen pretty much throughout the whole sampling period, which was over about a year in time. So these were then defined as these resident genotypes, whereas the other ones are transient. And that's important when you think of adaptation to an environment. You'd expect more thick clones to be stable. That's just ecological principle. Okay, so here's my pictorial of this model. So if you take a human stool sample and get the E. coli and genotype them, you're going to get many different types. The abundance of those types shown here, abundance on the Y, different genotypes on the X, might look something like this, where resident strains are very common and transient strains are only seen once. So what happens in the environment then when these stool samples get to outside the host? By the way, if you Google environment, this is the image you get. So what happens to these things over time? If there's no survival and there's no adaptation to the environment, then over time, these the rare strains will die, and the more abundant strains will just become less abundant. And so their distribution, or their original abundance distribution, would be a direct mirror of their fitness in the environment. Okay, so that predicts that if we go into a population of people, get the distribution of genotypes, sample the environment from those people, get the distribution of genotypes, those distributions should be exactly the same. Everybody good? All right. So what do we actually find? Here was a study of chickens and chicken barn floors. Okay, so we have a confined area where there's a host in their environment. A bunch of E. coli were collected from both chickens and chicken barn floors and genotyped with that electrophoresis that I showed. And this PCOA, principal coordinates analysis, shows the genetic similarity among all those E. coli. And all you need to know is the closer two dots are in this space, the more similar their genotypes are. Okay, so you see two main clusters, A and B. And so when you look at which ones came from the chicken, or the primary habitat, versus the barn floor, or the secondary habitat, and whether they belong to genotype A and B, you would expect, based on that previous slide, to see the same ratio in each of these boxes. And what was observed was a very non-random ratio, suggesting that strains that were most abundant in the primary environment were not the ones most abundant in the secondary. This would be, again, an expectation if some E. coli genotypes are more fit after they leave the host. Everybody got that? All right. So this sets up a hypothesis then that there are resident and transient strains not only in warm-blooded animal guts, but also in the environment. And so the question is, which environment should we go and look at to test this hypothesis? And at the time, there was a lot of work being done by a collaborator of ours in Michigan that was looking at the water and sand of public bathing beaches. Okay, and this is 6 public bathing beaches in Michigan. This is the water, this is the sand. They were measuring E. coli and another indicator of fecal contamination, enterococci. And you can see the scales here are different. But in the sand, you can get almost 10 to the third organisms per gram weight, 100 grams dry weight of sand. And if you average these numbers across all of the sites, there was a lot of E. coli in just that top layer of sand in these public bathing beaches. So suggestive that because of the difference in abundance in the water, which is presumably the source, and the ones that make it to the sand, that maybe this is a habitat where E. coli likes to live and reproduce. So this was the focus then of this project to study E. coli genotypes in this environment. So all Michiganders love 3 things: lighthouses, sunsets, and fresh, clean water. And this is a beach on the western shore of Lake Michigan where the sun sets. You can see there's a lot of people there. This is, of course, unless you're from Flint, where the water's not so great. And if you have followed this at all, it's what a mess, you know. And they're actually, this guy, Michael Moore, is a Michigander that's calling for the governor's head. So anyway, these are the jokes, people. So how are we going to characterize then these E. coli? You could do electrophoresis, but the first, luckily, there was a quicker way to genotype broadly these E. coli. And so I'm going to tell you about this technique. So if you build a dendrogram on a reference collection of E. coli meant to represent the diversity of the whole species, you can see 4 different clades here, and they're referred to as phylogroups. And those phylogroups have different letters, A, B1, B2, and D. Here, just color-coded on the tree. There was a PCR developed for presence-absence of 3 loci that would partition all the E. coli into one of these 4 groups. Okay, so just by doing a multiplex PCR, we could get a general idea of how the genotypes are distributed. And so this was the first technique that we used. Others have shown that pathogenic E. coli tend to be more in the B2 phylogroup versus some of these other ones. If you look at healthy humans, this is kind of the distribution that you get. This is taking into account about 100 healthy humans. You can see more A, a little bit less of B2, but you can start to understand what the distribution of these broad genotypes are. So we went into these 6 public beaches. We had samples spanning about 1.5 years in time from the water and the sediment, and we characterized 190 isolates. And here's what they look like across the beaches. You can see that B1 is the most dominant isolate that we found in in 5 of the 6 sites. And again, here are some distributions from different hosts. So right away, we knew that this looked pretty different than what we expect from humans. But shorebirds, ag animals, pretty similar distribution to what we found, suggesting that possibly these are the sources for these E. coli in the water. But these are broadly defined phylogenetic groups. They're not down to that clonal concept that I mentioned. To get down to that clone concept, we need to do more precise phylogenetic measures. And one such measure is multilocus sequence typing, where you sequence internal fragments of 7 housekeeping genes, concatenate those genes, and then build a phylogeny on that sequence. This was the dendrogram then from that sequencing effort. Again, this is 190 isolates, and we were able to define closely related lineages based on bootstrap values of the tree. And those are shown in these boxes throughout the tree. One of the clades, though, was found on a relatively long branch. The branch length was statistically significant. And also, the support for the node that connected all these lineages was highly significant. We named this clade ET1 because when we looked in the data, it was present at all the beaches over the whole sampling period. So it was very resident in the environment. And we said that this data supported the hypothesis that, that there were residents, and so it gave us a place to start. Since this original publication, other people have looked in the environment at E. coli. This was a study done by actually my college roommate that went on to do a postdoc at Cornell. And they looked at E. coli in the environment, particularly in soil, but they accounted for fecal deposition. So they made grids or transects out in the field. They characterized the E. coli from the fecal deposition and then looked in the environment across these transects. And what they found was that this ET1 clade and the E-Cort-D phylogroup couldn't be explained by the fecal deposition in their transects. So suggesting that either they were adapted to be there or that some other factor that they didn't account for in their study was responsible. So again, this supported our original finding, and they also found that these were very closely related clones. Okay, so just one more study. There's been multiple studies now on ET1, but this one I thought was interesting. This is one of the more recent papers on it. This paper used comparative genomics. So they sequenced a number of representative isolates, both of ET1, but also isolates from different hosts. And just to fill you in a little bit about E. coli and how genetically diverse it is, when the first 3 E. coli genomes were sequenced and compared, this was the figure in the paper. And I just wanted to point out that these 3 E. coli only shared about 39% of their genes. Okay, so we have a lot of gene differences between these strains. What this paper did, though, was say, okay, if we have, well, as an example, if you wanted to differentiate the genes or identify the genes as differentiated pathogens, like the uropathogenic or enterohemorrhagic strain from a non-pathogenic strain, you would design PCRs that target these genes. You would call those pathogen or pathotype genes, and these genes over here would define your non-pathogenic strains. And so this was the idea of this paper. And so they did this comparative genomic study. They found 6 loci that may be predictive of habitat, just based on comparative genomics. One of those loci is actually not a gene, but it's an allele of a housekeeping gene that defines the ET1 clade. So that was kind of cool. But anyway, so presence absence of 6 genes, you can have a 0 or 1 at each of those 6 genes. And so you can make sort of a 6-digit barcode. And that barcode then you can relate and see what the presence absence of those genotypes are in different sources. So just highlighting here, these barcodes were found to be enriched in fecal strains. These barcodes were found to be enriched in environmental strains. So the environments here are surface water and sediment. And quite encouragingly, I guess, 70% of all the environmental isolates that were positive for the ET1 clade only belong to 3 distinct genotypes, as based on this comparative approach. So that means that the ET1 clade, again, is very common in the environment, in sediment, and surface water, and they're very closely related. So that's 2 studies that supported it. A number of studies have now similarly found evidence for this. The term naturalized E. coli has been thrown around. I guess if nature means outside the gut, that makes sense. But anyway, the prevalence of these isolates are not trivial. We found in the beach sand study they were about 23% of all of the isolates. It fluctuates around that 20% value. So we're talking about increasing the accuracy of monitoring and decreasing costs associated with water treatment, potentially by 20%, just by identifying these from other E. coli that are enriched in fecal samples. Okay, so that's the story about the ET1 clade. I want to tell you another story that came out of this study. 190 isolates were originally considered in the beach sand study, but we actually had identified 195. And when we did our MLEE, or enzyme electrophoresis, and we did our multi-locus sequence typing, 5 of the E. coli really were divergent. And we didn't know what to make of that. And, well, my boss knew what to make of it. He said that they weren't E. coli. But about the same time that we were processing, so our beach sand paper came out in 2007. So about the same time this paper came out, and they found 2 representatives of a very divergent lineage, and these strains phenotypically match to E. coli. So there was some precedent that there are these oddball, or at least one oddball lineage of E. coli. And so we wanted to understand whether these fit into this lineage or some other lineage. So we called up collaborators that had diverse collections and screened those for these divergent representatives. So these samples included more Great Lakes water samples that had been done. I think there were a couple thousand E. coli in which 1 gene was sequenced, so we had a little bit of sequence data to go on. There was E. coli from pristine environments where humans were not. So one of those were Puerto Rican jungle, some areas in Australia. We expanded the sequencing to include 22 loci instead of just the original 7. The reason for that is these sequence data that generated this tree was different than the 7 that we used originally, but included in this data set. So now we could compare those divergent lineages directly to ours. So here's what we found. Here's E. coli, here's E. albertii, E. fergusonii, and here's their closest common ancestor, Salmonella. Our representatives clustered into 5 new clades of Escherichia. And so this was kind of exciting as a grad student because really this was primarily proof that when you go into the environment, a habitat that's not often sampled a whole lot for E. coli, and you do phylogenetic relatedness of the strains you find there, you discover whole new genetic diversity, suggesting that our perception of E. coli, both evolution and ecology, is very much biased by clinical isolates from warm-blooded hosts. Okay, so that was fun. That was cool. We published that, and then a lot of people kept asking, though, what is their abundance in hosts, and obviously that would take more sampling. So the way that you'd like to do this is find a freshwater habitat that is intensively used by warm-blooded hosts, something that you know has a lot of input, and then see if these cryptic clades are partitioned in the water versus the host. So anybody think of one of these? Anybody else not from my lab?
Toilet.
Well, the toilet would be good, yeah. But actually, we use the duck pond because it's a little bit easier to sample in your toilet. Maybe not. Yeah, that's true. Anyway, so the duck pond, as we all know, is home to, I don't know if we have a real accurate count, but I would estimate 70, 80 ducks. And, you know, they're very aggressive. This is the swarm. These ducks are about ready to attack an infant. So we keep these ducks around because I guess they're fun to look at, and we feed them. So I don't personally, but obviously some people do. So this is a great habitat, though, when you think about if you want to really push the system and say all that's going into there is just a bunch of duck poop. So everything in that system should be a result of that. And so we should see these cryptic clades, if they are getting into hosts, we should be able to sample them from the duck, right? That's the idea. But if we see them only in the water and not in the duck in this environment, then that suggests that this partitioning is maintained even in this intense pressure. That's the idea. Okay, so the question about density in the duck pond, I just thought this was funny. This was a mallard study, so we have primarily mallards. They had 2.3 mallard pairs per kilometer squared in these 39 lakes in Canada. This is just the average, but I mean, our duck pond's what? I don't know what square kilometer looks like, but it's nowhere near that, and we have way, like an order of magnitude more ducks there. Okay, so Escherichia then, that remain partitioned in the water versus the duck in this highly or heavily used system would add support to this adaptation hypothesis. Okay, so we followed the ducks around. We sampled poo after it hit the ground. And these pictures were taken in the summer, but we actually started in March. Pristine snow and things helped sampling. But then we also sampled from the water. And so just as a general summary slide, we had 19 duck samples over 10 different dates spanning from March to June. Water samples, we had 17 and 17 different times in the same time frame. And when we looked for the cryptic clades, we did find one in the duck out of 467 samples. And we found 9 in the water samples out of 377. So the ratio of cryptic clade to E. coli was not at random in these. They were very much partitioned into the water, suggesting that we were kind of on the right track in the beginning, that these cryptic clades at least are very much environmentally adapted. But what about partitioning? Here we can drill down into these different phylogenetic groups and ask, do we see patterns that might suggest their abundance in the environment? First of all, with the duck samples, as I told you before, there's resident and transient strains in warm-blooded animals. And so it's not unexpected to see 30 E. coli strains that you characterize from one poop sample to be identical. And the reason that we characterize 30, we actually try to characterize 33 per sample. That was our target. So there's a statistical calculator called rare events calculator. So if you draw 33 consecutive draws from a sample and you see the exact same thing, then the likelihood that there's something else in there is less than 5%. Okay? So if you want to be 95% confident that there are only B2 isolates in duct number 3, then you need to draw 33 consecutive E. coli, and they all need to be B2. Okay? So if there are other strains there, they're less than 5% abundant. So this gives us a look then at the E. coli in duct and how they're distributed. They're distributed very non-randomly. That's not the case in our water samples, where we see much more diversity. And that diversity, I highlighted A in red, so you can see that it's partitioned over to the later dates. And so showing this another way, in the water, we can just simply plot the percent of isolates per phylogenetic group across days, and that's kind of complex to look at, so I broke it down into 3 main patterns that we saw. Phylogroups B1 and B2 were present at about the same abundance throughout the whole sampling period. Whereas groups D and the cryptic clades, the one that other paper said were not explained by host and fecal deposition, were more abundant at the beginning during March, and then we didn't see them in the later months. Phylogroup A, on the other hand, we didn't see in the early sampling periods, but it tended to increase the warmer that it got or the later that the months got. And we don't really know what the ecological drivers are of these patterns, but we're really interested in that. Like, we want to go now and sample, we want to tag the ducts so we know which ones that we're repeat sampling. We want to go into the water and measure things that we know to be important for E. coli, like dissolved organic matter, the temperature of the water, pH, those kind of just general characteristics, and see if any of those variables trend and explain some of these patterns. So just as an aside to maybe point the finger at one of many possible mechanisms, a collaborator in Australia looked at cryptic clades. So the blue bars, burgundy bars, and green bars are cryptic clade strains belonging to different cryptic clades. They compared those to E. coli and the other known Escherichia species, E. albertii and E. fergusonii. And this figure is showing biofilm formation in rich media and in minimal media at room temperature and at 37 degrees. And what they found was that the cryptic clades tend to form more biofilms sort of across these different parameters, but most pronounced when glucose, in minimal glucose media at room temperature. They seem to be pretty separate, and also nutrient bereft at room temperature. So there's biofilm formation that might help explain. But the other thing they observed is when they decreased temperature, and there was probably, I forget how many strains of each of these clades were, or each of these species were in this, but there was a number of strains that they considered, and none of those strains other than the cryptic clade strains could grow at 5 degrees. The next growth of any other taxa was seen at 8 degrees C. So there seems to be some lower temperature growth in cryptic clade strains, which might help explain some of these patterns that we saw. If the cryptic clades were better at growing at cold temperatures, we would expect them in colder months. So we want to follow up on these to sort of solidify these results or measure other variables that might be important. So you might wonder why these strains have never been observed after nearly 124 years. So I just wanted to finish up on this and then summarize what we talked about. So the way that you identify microbes in the environment is you often do biochemical profiles. So indole production, the classic carbon, nitrogen, phosphorus sources. And so you can do this in commercial kits that come with substrates already available. You inoculate them with your cells and watch for either fermentation products or colorimetric changes or something like that. So we did that for, well, our collaborator did it for a lot of E. coli, so probably 800 different E. coli strains. And then we did it as well using the same kits for cryptic clades for E. fergusonii and E. albertii. So to try to understand what the biochemical or phenotypic differentiation between species are. And so what's plotted here in the open circles are E. coli, but not all the E. coli. If we plotted all the dots, it would just be a big cloud. So we only plotted the most divergent biochemical profiles for E. coli. These would be the 5% most divergent from normal, okay? And that's what's shown here with the open circle. One of the clade 1 strains, so 1 out of the 13 that we tested, was also in this 5%. The other 12 were just like E. coli and right in the middle, so we couldn't differentiate those. And so clade 2 came out, 5 out of the 6 clade 3 strains you can see, 2 out of the 4 clade 4 strains, but clade 5 isn't on here because it was identical. We couldn't differentiate it at all. And then you can see E. fergusonii in black and E. albertii in green. So we would expect these 2 other named species to be very phenotypically divergent from E. coli. And the reason we said in this paper that nobody's found these is because you have to do this biochemical phenotyping to identify them first, and they won't look anything different from what normal E. coli look like. Another study expanded this to 96 different carbon sources using the Biolog machine. And this is a complex slide, but just to summarize the main point, if you look at all E. coli versus E. coli, so Shigella are a little bit oddballs because they've gone through a lot of genome reduction. But if you look at E. coli versus E. coli, these red dots identify those comparisons. And so on the x-axis here is the genetic diversity between each of those pairwise comparisons. Okay? So E. coli span about, I don't know, from 0 to 0.3 on this scale. And you can see clearly that the cryptic clades shown in the diamonds are more genetically diverse. And that's what our dendrogram showed, right, that they outgroup from E. coli. So that's on the genetic partitioning. On the phenotypic partitioning, though, the cryptic clades are dead center in the phenotypic biochemical profile of E. coli. So this, again, supports our finding that you just can't tell them apart from E. coli if you're looking at biochemistry. But genetically, if you do phylogenetic relatedness, that's why you can, that's the only reason that, or the only way that you can tell them apart. But as said before, we're completely biased by our view of sampling E. coli. The standard reference collection called the E. coli reference collection, or ECOR, was generated on the allelic diversity of over 2,600 isolates, and not a single environmental strain was included in this analysis. Likewise, microbial source tracking is a common approach to identifying E. coli that we know or we think are associated with a certain host. And you look for those in the environment as indicators of pollution. But this requires a large effort in sampling different isolates. And if you sample more isolates, you have less money to sequence, okay? And so there's a trade-off then for using fingerprinting tools that all you get back is same or different versus sequencing-based approaches where you actually get genetic relatedness. So a lot of the characterizations of environmental E. coli that have been done only tell you whether they're same or different. And so you have no resolution to say whether that's a cryptic clade or not. And then finally, temporal dynamics. If these things are resonant, the only way you're going to define a resonant is by seeing it over and over. And so how you sample is very important. The 2 main tenets in microbial ecology is presence, absence, and relative abundance. And those are what we focus on a lot in these cross-sectional studies. But we never understand really by those samples, by those types of samples, what that diversity does over time. Okay, and that's very important for our definitions of these adaptations. And a lot of samples out there, if you really read in the methods, are just simply samples of convenience. And these don't really represent a habitat all that well. Okay, so as promised, what's the impact? Why do we really care? I mentioned this before, but if we increase the accuracy of defining what fecal indicators are, then we can decrease costs associated with water quality, cleanup, bioremediation. But we can also use E. coli, these environmental strains, then I think as indicators to help us understand what the total microbial community is doing, especially the ones that are facultative like E. coli in the environment. We know that there's a lot of temporal variability where bacterial counts peak in northern climates like ours in the summer months, and then they go back down in the winter months. There's a lot more carbon available for them. But also, this third point I think is important too. Now that we know that these things are more adapted to outside the host, they don't really find themselves in host a whole lot, they might be able to indicate what our human impact is on the environment. And what I mean by that is if we look for gene flow, things that are important to us like resistance factors, we actually did some comparative genomics on these strains. So shown here in a circle diagram are E. coli. Here you can see the A, B1, B2 groups. Group E is kind of an oddball group. It's within the old group D. But anyway, what these lines, these connections are between the cryptic clades 1, 3, 4, and 5, what they represent is recombination events. Okay, and A is the core genome, or the genes that are shared among everyone, and B are the ancillary genome. So genes like antibiotic resistance and genes important for, environmentally specific functions. And hopefully you can see that there's partitioning in core gene recombination, where this is only showing recombination between cryptic clades and E. coli, but one of the cryptic clades recombines a lot with E. coli, versus these other clades, 3, 4, and 5, only seem to be recombining with themselves. And this is core genome, okay? But this suggests that C1 is actually more closely related ecologically to what E. coli is. And when we go and look at these strains, these are actually enterotoxigenic E. coli strains that have never been sequenced before. So that was kind of cool. But the main point is what's happening with the ancillary genome. There doesn't seem to be any patterns here that I can see or that the statistics that we ran on it can find, which means that you're equally likely to exchange a gene between cryptic clades and E. coli as you are cryptic clade to cryptic clade. Okay, so what that means is that these E. coli in the environment could exchange genes with E. coli that we propagate through animal husbandry or wastewater or things like that. They can pick those up. We can identify then the rates of gene flow throughout the environment in a very ecological context. Okay, so future directions. We want to understand in the lab what genome factors promote this residence and transience, both in the host and in the environment. One of the observations in 1981 that I keep coming back to and dream about at night is this. So shown here in different blocks are residence strains from that person that I showed you before that was sampled through time. Each column here is a representative of a resident strain. You can see here this one's labeled 2A, B, C, and D. So these are the exact same genotypes, but these profiles are the plasmids in these genotypes. So taken 4 different time points throughout the year, the same clone with a very different plasmidome. So what functions are on these plasmids? What dictates which plasmids are selected and which ones go out? You can see that diversity is very different from genotype to genotype, from resident to resident. This one doesn't have much change. There is some. This one and this one have very whole-scale changes in their plasmidome. So these plasmids, I don't think anybody's looking at this and how that influences how these strains set up residence and which plasmids are picked up and propagated to the population. So I think this might be a key future direction. Okay, so I'd like to acknowledge the people in my lab. All of this work, for the most part, has been supervised by my lab manager, Sue. Where's Sue? Did she leave? Oh, there she is. But supervision of only undergrads. So no graduate students or postdocs have done this work here at MSU. We've had support from the USP. My collaborator, Liz Alm, has an undergrad in her lab that's been doing a lot of this research as well, as have other collaborators that have helped get this project off the ground. And apart from USP, the only other funding source was MSU Startup, so it's kind of appropriate that we use the duck pond to study this concept. But with that, be happy to take any questions.